So I'm going to actually start us off by everybody introducing themselves because we're a small enough group that we should know everybody. So I'm Susan Hayflick. I'm a medical geneticist. I work at Oregon Health and Science University. And yeah, some members of the OHSU team here as well. So maybe we could just go around this table first. Introduce yourself.
Yes, I'm Lex Peters. I'm
Lex.
from the Netherlands, and I'm the grandfather of a son with MPAN, but we also represent other families in the Netherlands with different diseases. Thanks. And it's a great pleasure to be here.
Yeah, thank you.
To meet all of you.
Yeah.
I'm grateful for that.
My name is Allison Gregory. I'm married to Rex, so my story is quite similar. So I think that's it for now.
Hi, my name is Cheryl Lamus. I'll just stand up for a second. I have 4 children, 3 of which had the MPAN that we're going to be talking about today. 2 have passed on, but 1 is still with us. So I'm interested to whatever is new about this. I've met Dr. Hayflick almost 20 years ago, though, and I think they're doing a great job. Keep up the good work, motivating other people.
Thank you.
I know you can't do everything yourself.
You're one of the rare ones who've been to all 10 of the meetings.
Well, only 9. I missed the first one.
But it's families like yours that sort of kept us focused on not just the genes we'd found, but the genes that hadn't yet been found. And the diseases that still need to be
I never thought you'd
discovered.
find it, honestly. I
Well,
was
we didn't.
surprised.
The Munich group did, but that's...
Right, exactly. I
that's
didn't
fine.
think it would be found.
Yeah.
Well, since our story's the same, I'm Michael and Peggy Wickert, and we're here for James. He's our second son, and he has MPAN, and we've tried to figure out, and we're grateful for, all that you guys do. So, again, to keep up the good work. Thanks. Hope that did it.
Yeah, and Peggy.
Yeah,
go ahead. Yeah.
okay, thank you.
Nowadays, I'm studying coenzyme 8 to autophagy 2, unknown mitochondrial protein, everything is going to be.
I'm Allison, I'm from OHSU, I'm a research counselor. I think you all know me, so I'm going to stop there. One big, and dad like that. I couldn't endure. Kathleen Ayers, Gigi's mom. Medical psychologist, education administrator, independent researcher, and nutritionist.
Jim Bourgeois, Gigi's dad, and chair of psychiatric academic medical center here for Gigi.
My name is Kevin Ozakowski. My 2 kids, Kieran Ann and Kevin Paul, have MPAN. And my wife Chris is here. We're fortunate to love as well. So, pleasure to be here. Thank you all.
I'm Dick Ritter. I'm a pediatric neurology resident at Cincinnati Children's.
Jen Wilson, pediatric neurologist at Oregon Health and Science University.
Thank you.
I'm Sunita McIntyre, I'm a pediatric neurologist in Ottawa, Canada. I'm Laura Kosher, and I'm a pediatric neurologist and moving disorder specialist at Children's National in Washington, D.C.
Thank you.
Allison Gregory, study for
Thank you. Dale, tell us who you are.
I'm Dale Ward, I'm just here to fill. I have a grandson with PCAN.
Yeah, he's helping out with the AV. So you noticed the families, you noticed that we have a cadre of early career child neurologists who have been invited by the Oregon Family Organization to both participate in these sessions, learning sessions, to hear from you. And then this afternoon, they'll be in a kind of an intensive training session examining some of your family members and also others here at the meeting, trying to get a pretty intensive snapshot, mostly examining, but also talking to you about your family story, examining your family members. So we have an hour. Now we have 50 minutes. And we have, this this could be a lot of things to talk about. It could be not so much to talk about. I'm not going to stand up here the whole time and talk. I'm going to just frame some of the stuff to talk about, and then, you know, ask for questions. And so I'm going to start off updating you on research. And Dr. Ayuso from Munich has provided an audio slide presentation, basically a video, that I will run. I haven't watched it. I don't actually know how long it is. I think it's about 10 minutes, but if it turns out that it's 45, I might stop it early. I also, looking at the slides, it looks pretty intense scientifically. And so some scientists are really good at hitting the right mark in speaking to families, bright families who aren't themselves scientists. Let's see how she does. I'm going to start off by inviting Allison to talk a little bit about the work that we've done over the past, maybe, actually it's been a lot longer than that, but at least 5 years for the dominant MPAN. So go ahead, Allison.
So, I think the last time we saw y'all 2 years ago, we were just starting to talk about writing a paper and sort of putting together our data about dominant cases of MPAN. And we were still collecting important information at that point. We hadn't totally built our case. Even prior to that, we had one family in particular where, like, every year we'd come back scratching our heads going, there's no other way to explain this. It's just, but the genetics weren't quite working out for other reasons, and there was a deceased family member that was hanging this up. And I guess I should back up and say what's unusual about this is when MPAN was first reported, when we were first looking at the families in our repository with MPAN, it was recessive. And it was pretty clearly recessive. And part of the reason it was clear was there was a common Polish mutation, and that one is recessive, meaning both parents have to be carriers. They pass it on to an affected child who gets both copies. It's pretty odd, not unheard of, but pretty unusual to have a disorder that can be both dominant and recessive. So that's what we were proposing. So we had to really have good evidence for that, which we finally, with some collaborators in New York, found a second family with a big pedigree, and then we had two, big family trees where you could clearly see it going in a dominant fashion from one generation to the next. And then we did some additional work with another collaborator in Germany, different from this group that we're going to see a video from, and sort of built our case and wrote a paper over a period of time, submitted it. They're not always accepted the first time, so we ended up kind of I'm putting that lightly. We reworked it a few times, added some things, subtracted some things, but eventually it was accepted. It was actually just published probably 2 weeks ago. So I can send a PDF. I don't have a paper copy here, but I can send a copy to anybody who would like that. But it's really important. There it is. That's a page out of it. Those are our two dominant pedigrees.
So just each of these is a family tree. The circles are females, the squares are males. These people are siblings. That's their mother. That's their uncle, their aunt. But you can see that a grandparent, a parent, a child, and a grandchild have MPAN. So it's traveling through the generations, which is not how it's traveling in your families. It's traveling just in your offspring, in your children.
Yeah, and neither of those families happen to be here this year. But some of the implications, part of the reason we want to get it out there is it's really important in terms of clinical labs interpreting test results and writing reports for newly diagnosed cases. Because before, if they just saw one change in the MPAN gene, they wouldn't necessarily call it as MPAN. They didn't know how to interpret that because it's supposed to be recessive. You're supposed to find 2 copies, and what we found is finding one, especially certain types of mutations located at the end part of the gene, really is sufficient to give you MPAN. And the other thing that's important, or that this helps prove and helps to change the test interpretation, is for people who are tested with their parents, if their parents both test negative, but you see one new change in a child, that's another way that dominant disorder can happen. It can happen as what's called de novo, or a new change just in that person. And now that type of result would also be more clearly interpreted as being MPAN.
That's great, thank you. So this is one of these papers that, you know, in the grand scheme of papers is actually quite a small one, but its impact on families affected by MPAN, where the disease is dominant, is profound, I would say, because a month ago, the laboratories would not have made the diagnosis at MPAN, and now they will have confidence being able to make the diagnosis with a single mutation of MPAN. So it doesn't change the stories for those of you who have known recessive MPAN. It doesn't change the story for your families. It doesn't change the story for your healthy children and their chance of having a child with MPAN. None of that has changed. This is, as Allison said, when the mutation is in the very, very end of the gene, for good biologic reasons, it leads to this dominant MPAN. And so it suddenly, labs around the world, when they see a single variant, will have the tools to ask, is that enough to cause MPAN? Is it in the right part of the gene? Is it the right kind of change? Those sorts of things. So it has an immediate, we say, clinical impact or impact on care. Okay, so the other stuff that's going on that is really sort of out of view but is quite exciting to me. So there was a call for funding for MPAN. This just very recently, I think the submission date was just a few weeks ago. And some new scientists who either have been quietly working on MPAN or haven't been working on MPAN before submitted proposals or started to think about submitting proposals. So there was one early career scientist in Arizona who has some really good ideas for a new disease model. So remember, the way that we figure out a disease, the way we explore and investigate a disease is partly on your family members. We do take samples and we study the cells in the laboratory, but we also try and create animal models because they're something that we can manipulate and try drugs on and do things with that we can't do so well in cells in a dish, and certainly can't do legitimately on your family members, wouldn't do on your family members. So there's a fruit fly with MPAN, and now there's an interest in making what's called a zebrafish, which is a special kind of fish where you can sort of visualize changes quite easily. There are a lot of tools for studying zebrafish, and there's an interest in making a zebrafish model, which if this scientist can get, I think, a little bit of funding and some traction on, could happen actually very quickly. It takes a matter of months, really, to create that animal model, and then a number of months and years, probably, to study it. But that's potentially quite a powerful tool. There's also an interest now, so the story when the MPAN gene was found was that there was no MPAN gene in mice, and so we couldn't make a mouse. Mice are usually what we go to as animal models, but now there's an idea that maybe there is an MPAN gene in the mouse, and there's an interest in exploring whether that can be a good mammalian model, right? Because we always want an animal model that is both fast to reproduce, but also, I wouldn't say similar, but closer than zebrafish or fruit fly. That's mice. There's also, and this is something that's quite exciting to me, there's an interest now in making what are called pluripotent stem cells. So we can take a bite of skin from your family member, or from you, and we can turn it into a stem cell, and then we can treat it in such a way that it becomes a neuron, it becomes a brain cell. So what used to be a skin cell in a person was taken back to its infancy, if you will. It becomes a stem cell that can become anything, at least in theory. And then it's turned into whatever cell we want it to become. And in this case, we want it to be a brain cell, because we don't have other easy ways to have living brain cells in a test tube, and this is one way to have that. So there's an interest in making what are called iPSCs, induced pluripotent stem cells, iPSCs. There's an interest in making iPSCs and making neurons in MPAN, and what that then gives you is not just cells in a dish, but brain cells in a dish, which is a very powerful tool for studying the disease process, being able to ask questions in the relevant disease tissue. Those models are essential tools to think about how to discover, screen for potential therapeutics. Many of them require a basic understanding of the biology of the disease, but not all of them. So if you're going to come up with a rational therapeutic, a therapeutic that makes biologic sense given what you know about a disease, you have to know something about the disease. And in MPAN, MPAN is a really tough nut. It is really tough. Still, so when was the gene? The gene was reported in 2011. It was found in maybe 2008, 2009, somewhere in there. It's been almost a decade, and we don't know what the gene, so genes are instructions, right? They make proteins. We don't know what that protein does. We know it's associated with mitochondria, the energy producers in the cell. We know that it sits in a membrane. We think we know that it doesn't act alone, but it acts with itself. It actually joins itself, at least to be a dimer, a two molecule, but it might actually be bigger than that. But what's it doing there? We don't know what it's doing there. And so when it's not working, we don't know what's not working, right? So you've got to, I mean, you can make your own analogy and figure out, well, if you don't know why there's water in your basement, or why something's not working in your house, you can't fix it. There is one approach to therapeutics that doesn't require that you know the function of a gene or protein, and that's gene therapy. Well, we'll just get a new house, or we'll just get a new plumbing system, right? I mean, it's not a great analogy, but we'll just put a new gene in. And so there's an interest now, if we can get good models, particularly a mouse or some other kind of mammal, probably a mouse. If we can get a mouse model of MPAN, that's an if, that's a big if, and that's a lot of work. That's not a couple of months of work, that's a couple of years at least of work. Then I think, and some of this work can be done in the iPSCs that I was talking about, but it would eventually need to go through probably a rodent model. Gene therapy, there's an interest now in exploring the feasibility of gene therapy in MPAN, and part of that is that there's a recognition that gene therapy technology and discoveries have advanced really quite remarkably in the past 3 to 5 years, whereas our knowledge of what the MPAN protein is doing has not sufficiently advanced. So after we introduced ourselves, a new person, actually a couple new people joined us, so I'm going to ask you guys to introduce yourselves, but point out that L'Oreal, in the back waving, is a scientist who works on gene therapy. And she herself is not specifically working on MPAN, at least she wasn't yesterday. So could I ask you to introduce yourself? Yeah.
My name is L'Oreal Early. I'm currently a postdoc at the University of Chapel Hill's Gene Therapy Center, University of North Carolina, Chapel Hill. And I work in AAV-based gene therapy, which is a viral vector-based gene therapy. So in that case, we would be able to put the gene into a virus that's been modified so it doesn't cause disease, and we can put that information drivers for gene function, and that's worked really well so far in several types of diseases. But I just learned about MPAN now, so.
And next to her, somebody, the knitting parent.
Yeah, I'm the knitting parent. My name's Stacey Estrada. My 5-year-old daughter has PCAN. So I've had the interest in genetics. So I've been sitting in on most of the sessions and just learning more about the community family.
Welcome. Okay, so that's my very fast, very broad overview. There were some questions submitted ahead of time about sort of some clinical questions, and I thought we could address those briefly. And then, again, the time, we have just the hour till 9. It's 8:36 now. So I'm going to jump in and talk about diets, supplements, and interventions for mito support, and then maybe open it up to questions. to the best of my knowledge today, there are not dietary supplements or dietary changes that I could make sense of as an intervention in MPAN, and again, largely that's because we don't know the process that the protein is involved in. It is the statement of its involvement in autophagy is a very broad statement. Autophagy is a very complex process, and it may or may not be the only process that the protein's involved with. And so sometimes there are mitochondrial supplements that can be taken for specific mitochondrial disorders, and some of those may have been tried in your family members, but to the best of my knowledge, none of them has a specific rationale. And although I think there's low risk in trying them, something called a mitochondrial cocktail, usually they're focused on trying to rescue the energy producing function of the mitochondria, and as Arkangela's student commented, the energy production is not impaired in MPAN. So these are things like alpha lipoic acid, and again, I'm not recommending these, I'm just giving you examples. Coenzyme Q. Some of these are things you can buy at a nutrition center. They're not prescribed, they're not drugs, they're called nutritional supplements. And again, some of them may have a rationale, but to the best of my knowledge, none has been shown to have even anecdotal benefit in MPAN. So I'm not recommending them. I do want to just make a quick comment on iron. So we're here together at this meeting because of the term neurodegeneration with brain iron accumulation and this commonality. So all of our affected family members have not just iron in their brain, excessive iron in their brain, but they have a process that is a change over time, a loss over time of function. The iron is not the problem in any of the disorders represented at this meeting. So it's a sort of a side effect of the disease. I think of it as collateral damage. The accumulation of the iron is a bit of a marker of the disease process. And whereas it may itself be contributing to the disease process, it may be adding on to the disease process, we don't know. If we could take all the iron away, I believe there would still be a very significant and very problematic disease process. And so my own group we've never focused on the iron and getting rid of it. Some of the other groups have spent more time and energy on that, and you know there's been a trial of a chelating agent, a drug that sucks up iron and helps your body excrete it in your urine. And should we try that in MPAN? You could make a justification. There's not something else to try. Maybe let's just try it. Some families maybe have. You've talked to your physicians and advocated to just try it. There hasn't been a controlled trial. I think that would be difficult to do anywhere but in Europe, where there's a concentration, because there's a common mutation. There's a concentration in Europe of more people with MPAN than anywhere else in the world. Mostly in Eastern Europe, mostly around Poland and Germany. So I think if anybody's going to do that trial, it's probably going to have to be done there. But I haven't, to the best of my knowledge, that's not something that's in the works or in the plan. To do that kind of a trial is a huge undertaking. It's several $1 million, even with a compound that's already FDA approved for some things. But we know how to do it. We did it in PCAN. The results of that study should be published sometime in the next few months. And the outcome of that study is if you really grind on the data, you can see a difference in the data. Did families see a difference in their family members? I'm not convinced. So my take home from the PCAN trial of that iron chelator is that it may have some statistical benefit, but it was not very convincing to me. And so I still take a middle-of-the-road approach. If a family wants to try chelation, I try and direct them to somebody who can help advise them, and if they don't want to, I fully accept that.
And I would just add, for those of you who haven't looked into the chelation protocols, so we'll talk to your providers about it. It does require weekly blood draws for a period of time, because there is it's a very, very rare risk or side effect of this drug, but when it happens, it can be devastating, which is it can make the white blood cells go down to a dangerous level and make the person very vulnerable to life-threatening infections. I don't want to overstate that risk because it is an exceedingly rare complication, and if it's picked up quickly, you stop the drug and the white count comes up to normal. But what that means in practical terms for families is that they have to commit to getting weekly blood draws for their family member who has MPAN. And that may be not such a big deal for some families, but for some families it's a real burden. And so, you want to go into something like this with eyes wide open. As Susan said, I think in the U.S. this is always probably going to be done on a case-by-case basis with a family working with their provider and getting advice from outside. But we want to make sure, because we don't know of any benefit, that there is no harm. And the harm is a burden of weekly blood draws, potentially a cost associated with that, and this small but real risk of physical harm from a drop of white blood cells. We just want people to go in and eyes wide open. We take a kind of an agnostic approach when we see people in the clinic and say, if you want to do this as a family, if you want to try mitochondrial supplements, if you want to do things, just always consider that we're supportive of what you want to try for the most part, but always consider the risks and potential benefits, the evidence for benefits. So that's what we sort of say is a guiding principle. And I apologize for being like, I was on call.
I'm going to open us up. We've got about 15 minutes left to see if you have questions, and then, or if you just want to tell us something about your family member. Yes.
This may be the only time we would have more doctors and researchers in the room than families with MPAN in their families. So great question.
I guess you talked about, taking a sample and then being able to convert it and all of that. Is that active? Are you doing that?
So we are set up to take skin biopsies here at the meeting.
Okay.
We haven't been sort of
Diligent?
well, we haven't been out kind of, grabbing people by the collar and pulling them in. We've been announcing. So yesterday we met with BPAN, PCAN, and PLAN, and we announced it there. We are here again today collecting samples.
Okay.
If you have family members from whom we have not yet collected a sample, we would be interested in doing that. We do have cell lines in the laboratory from maybe 5 or so families already with MPAN, including, I'd have to check. I don't know whether we have your family members or not. We might have taken a blood sample and done a lymphoblast culture. Yeah.
Maybe a blood sample.
Yeah.
Okay.
Okay.
I don't know that Jim
Yeah,
was
but
able
not a
to make
skin
it.
cell. Yeah.
Yeah.
Okay.
And then I think that there'd be one other thing that I really, so all of you that are here for neurology, and I'm not going to drag this out, but, it's really critical. Jim was in the ICU for 6 weeks. A neurologist came in, didn't know what was going on. We can accept that, because it is so rare. But what we can't accept is when, people, and I'm so grateful that you are here and listening, because the doctor that came in did not listen. We explained a lot about the disease and that. And Jim's getting Botox in his forearms so his hands don't move. He has optic nerve atrophy. But yet after we explained all of this stuff, and he even went so far as to tell us, that there's a cure out there for MPAN. And we said, wow, you're really misleading us, and you know, all of that. And so anyway, he put his fingers into Jim's hand and said, squeeze it, squeeze it. And Jim couldn't do it, Botox and all. And waving his hand in front of Jim's face, and he couldn't see it after we had explained all of these things. And so in with saying all of that, please keep in mind, and we're so grateful what these poor people are going through and how they are trusting in, you know, your knowledge and your care and doing what you do. So God bless you for doing it, and I hope that I encouraged.
Yeah, it's hard to have confidence in a doctor who has undermined their own abilities. Yeah. Thank you.
It's a good principle for us providers to know that when you walk into the room with a family who has somebody in their family with a rare disease, they are usually much more of an expert than the person walking in the room. It's a rare instance where the provider knows more about the disease than the family does. And so the people who come here, unfortunately, are not the ones that need to hear that message, right? We're all trying to learn more about rare diseases, but most providers really need to hear the message. So thank you for sharing.
And so when we went into Select, which was a secondary thing, there was a doctor there that actually walked into the room and said, I'm going to be honest, I don't know anything about this. And I shook his hand. And I said, thank you. Because the honesty is what's going to cure. The honesty is what's going to discover, you know, and all of this stuff. So, thanks.
There's a...
We work together.
There's a...
Exactly the same thing.
There's a carving in the stone. So I work in I've worked in the same building for 27 years, 28 years, and there's a carving in the stone outside that says the obstacle to discovery is the illusion of knowledge. And I walk past that every time I go in and out of the building, like as if I need that reminder that many times.
Don't forget, Hayflick.
What other questions or comments do people have, including from the physicians in the room? If you have questions of the families, you should feel free to ask. Can I just ask each of the families of individuals with MPAN, how many years was it, with the understanding that for many of you, your kids are adults, and so there was no MPAN gene for many of the years that they went without a diagnosis, but how old were your kids when they got a diagnosis?
Well, James was 8 years old when he was patting the ground. That was the optic nerve atrophy, but until he was 16, he didn't have a NBIA diagnosis, or when he was 16, he got that. But then...
In 2011.
2011 was the MPAN.
So with the gene discovery, probably we got you all, or pretty quickly.
Right, but still, we went down roads, and you know, with your information, you know, like Back to the Future, there are no roads. It's, you know, we were directed down different things that were pointless. Yeah, that was tough.
Yeah.
Probably Becky was about 18, where Bruce might have been 16. That's when we just noticed awkward gait. We really didn't have any symptoms up to that point. I can look back on some videotapes and see some things that I understand now were there, but they didn't really have any deficiencies up to that point.
Okay. Yeah.
And each of them took a different path as far as, like, you know, a plateau, and then it's amazing, you know, even 3 that had the same.
Yeah, even within a family.
Within a family.
Yeah.
The rate of deterioration was different.
Yeah, that's an important message. We also see that in PCAN.
Mm-hmm.
We can see really quite different disease courses in siblings and cousins and extended family. Yeah.
Kind of similar, 15, 16 pairs.
Yeah.
Yeah, my kids were about 16, 18.
Uh-huh.
And so when you gate, you know, and the processes and all that.
Yeah.
I mean, at least you had something to go on.
Yeah.
And we know there's something there.
Right. Okay.
Yeah, our daughter was diagnosed at 16, About 4 years or so of increasing gait and balance. Problems, cognitive problems before that. That
Yeah.
was more nonsense.
Yeah. So these are not babies, they're not even young children necessarily showing up in your clinics, but showing up with sort of new changes in adolescence maybe, when everybody's having changes. Yeah. So I think recognizing MPAN early can be quite challenging. It's one of the disorders that is in the differential diagnosis of juvenile ALS. So we've had a handful of individuals come see us in Oregon who had a diagnosis of ALS, and then an MRI was done, and it was clear that there was more going on. So the MPAN MRI is quite distinctive in many individuals. It's to my observations, I would say in most people with MPAN, it's virtually diagnostic, kind of like PCAN, but you have to know. So we look at MRIs all the time and offer that. So the other thing physicians who don't know about MPAN can do is they can reach out to those who are more knowledgeable, and we're always open to that. So you all know that.
Yeah, I'm sure you do.
Yeah.
Given the time frame of diagnosis that has been discussed, do you think there's any role for neuromigration, synaptic pruning, or hormonal changes in the expression?
That's an interesting question. So you're asking the question, why don't we see it in babies and young children, but we see it in adolescents? Is it a function of those normal processes going in a different way? Well, maybe. The piece of information that I use to make me think probably not is when a person with MPAN dies and the family donates their brain, and we have an opportunity to see that brain, to examine that brain, which is something that we've worked hard to work with families to help them sort of see the value of doing that, if that's consonant with their own personal values. It's been an extraordinary teaching gift to not just us, but to others, because we share tissue with others. What the brain pictures show us is even in the teenage years, is a massive accumulation of proteins that normally we wouldn't see in the cells. So particularly alpha-synuclein. Alpha-synuclein is a protein that is people don't know if it's the cause of or the reaction to a disease process, but it's almost synonymous with Parkinson's disease. It's the thing that we see in old brains with Parkinson's disease, and also something called Lewy body dementia. People with MPAN have more synuclein than any other disease. And so I actually think it's the accumulation of that synuclein, or whatever the process is that leads to that accumulation, that just reaches a threshold by adolescence. Although there are some younger people.
For some reason in this room we happen to have the
Yeah.
onset, but there are, I can think of 6, 7, 8-year-olds.
Yeah.
And people who've had cognitive issues from pretty early.
So intellectually disabled people, maybe people with more of a movement disorder, dystonia, some behavioral sort of neuropsychiatric problems in childhood. That's kind of the phenotype of MPAN. Yeah. So we do have a little bit of a skewing of representation towards the older people. I actually think it's the accumulation of synuclein that is starting to kind of overcome the normal processes. I think that synuclein starts certainly after birth and probably before birth, but it just reaches a stage where now the normal processes can't beat it out. Just like in Parkinson's disease, it's going on for decades before you actually see the person showing up with a tremor, but it has reached a stage where the body can no longer compensate, the cells can no longer compensate. That's how I think of it, but the factors that you're asking about may well be contributing as well.
It just highlights that we really do have to do more work on MPANs as well. We don't understand about protein, about basic biology of the disease. It's surprising to me
And I say that only because the dominant disease is generally later in onset than the recessive disease, although in the families in this room, you're seeing kind of a later onset phenotype. I don't know that we have really good insight into that because there is some information about regions of the gene and their functional significance, but still quite a lot that's unknown.
We have looked at those, late onset rapid cases, and their mutations, like between those families, they're not.
Yeah, they're not.
Similar.
There's nothing distinctive.
If you were ever going to want to track down genotype phenotype correlation, you would think if you look between them, somebody who has like totally normal cognition, neurologic exam for most of their life, and then suddenly in middle adult life they go completely to hell versus an earlier onset, first decade of life child with MPAN. Do you think we'd be able to get some clues from the genes there? Just it's weird.
I used the wrong term before when I said diligent. I'm sorry. I apologize. But the other thing is that James is totally cognate. He knows what's going on. As a matter of fact, I can tell a joke in a room, he'll be the first one to laugh. And it's just amazing. The thing that he has is struggles to communicate.
Sure, to express himself.
Yes, absolutely. So he is so trapped inside of his body. But that's so working.
That's a really important point for the whole care team to know about, so that nobody's talking about him with the assumption that he's not picking everything up.
Right, talk with him because he knows.
I believe though he has early onset because when he was 8, he was diagnosed with optic neuritis.
That's when
so I think
things
he has
start to
to
fall
work
apart.
Are the age groups like to be parallel with the PCON classical and atypical?
Yeah, so the question is, are there age groups sort of in parallel to the idea of classic and atypical? We haven't really named it in that way because there's a huge spectrum. There's a huge spectrum in PCAN as well. I don't know, Allison, do you have any thoughts on that?
Because the population's just smaller, we haven't tried to separate them.
So we're up on the hour. The training physicians in the room will be in a session this afternoon learning about MPAN with hands-on. But most of us are around through a significant part of the meeting and would be happy to spend time talking if you have further questions. So I'm going to wrap us up for the formal session. And I do know that there's that Patty's got an agenda and a sessions going as well this morning, so I want to respect people's interest in participating in those. Yeah, thank you all.
Okay, thank you.
Thank you.
Thank you.
Thank you very much. Thank you, of course.